Well, welcome. Welcome to today's webinar. My name is Libby Kennard, and I'm one of the program coordinators with Parkinson's Resources of Oregon and based in Lane County. PRO has 2 regional offices. This one is in Eugene, and we have a new one in Bend to serve Central Oregon. Also this fall, our headquarters moved to an expanded facility in Beaverton. So it's pretty exciting to be able to grow so that we can provide more opportunities for classes, groups, and personal support. Upcoming, we have an educational program that's going to be in Springfield on November 16th. It's for care partners to learn more about how to be prepared for the advanced stages of Parkinson's disease. If you want to learn more about our services, just give our office a call. So for today's webinar, we are talking about research developments in Parkinson's disease. It's going to focus on the evolution of Parkinson's disease, medications, alpha-synuclein mutations, genetics, stem cells, and some research on the gut microbiome. You will have an opportunity to submit your questions as we go. So let me tell you how to do that. On your screen you have a control panel, and there's different panes. You'll see a question pane, and then either a plus minus sign or an arrow that you can click that will open that. And type your questions into that box and hit send. At the end, we will save time for questions, and also some of your questions will get answered along the way. So now let me introduce our presenter, Dr. Richard Rosenbaum. He is a neurologist at Portland Parkinson's Program with the Oregon Clinic and Legacy Health. Dr. Rosenbaum also serves on our board at Parkinson's Resources. He's an expert in the treatment of Parkinson's disease and is the author of the book, Understanding Parkinson's Disease, A Personal and Professional View. So now I'm going to turn this over to him. Thank you for being with us today. And here we go.
Thank you, Libby. Thank you, Libby. I've never given a webinar before, and I was a little bit concerned about talking without being able to look the audience in the eye and knowing what they were understanding. So I asked my 8-year-old grandson for advice because I wanted to talk to someone who was a digerati, and he said, well, give them a quiz at the end and then you can find out if you taught them anything. And so I want to assure you there's not going to be a quiz, but I'm delighted that you have a chance to quiz me. So I really rely upon your questions, which Libby's going to be sending to me as I speak, so that if I'm a little bit off the mark, that I can clarify things for you. There's so much going on with Parkinson's disease right now. There were 4,400 research articles written on Parkinson's last year that obviously we can't talk about them all, and I'm just going to give you a smattering of what's going on. I hope I've picked some exciting things. At the first World Parkinson's Congress, which was about 2007, Michael J. Fox was giving the introductory talk, and he looked at the number of research articles that were there, and he said, but I can't tie my shoes any better. And I want to start by just talking about some medication changes that have occurred in the last few years that really do help patients move better and live with their Parkinson's better. So this is the 200th anniversary of Parkinson's description of the illness and the 50th anniversary of Coates' introduction of DOPA therapy, which really changed what we could do for Parkinson's disease. And I want to just mention a few things that have come up in the last 3 years for new drugs that are the product of research that have changed the way we deal with Parkinson's for appropriately chosen people. And I want to talk first about drugs that deal with the problem of motor fluctuations, which are also called on-off. So when you have Parkinson's initially, and you start to take drugs like L-DOPA, they usually work in a very smooth fashion. You can take it 2 or 3 times a day and have it work around the clock. And many people, after they've been on the drug for 3 or 5 or 10 years, find that instead of having that smooth action, the drug turns off after a certain number of hours. Maybe it's off when they wake up in the morning. They take their first dose. It may take half an hour to kick in. As it kicks in, they may get extra movements called dyskinesias, and then as it goes down in the blood, they have increase in their Parkinson's symptoms, the slowness of movement, the bradykinesia, the rigidity, the stiffness, the tremor, as well as increases some of their non-motor symptoms. For example, for some people, pain or anxiety can get a lot worse when they're off. So dealing with that on-off is a real challenge, and there are a number of ways we do that. One is to make the L-DOPA last longer, which we do with drugs like selegiline, entacapone, rasagiline. They're now slow-release kinds of levodopa, carbidopa. There's some disadvantages to each of those, which is why we only use it for people who are really having problems. Deep brain stimulation can be extremely effective for dealing with the motor fluctuations with the on and off. But the things that are new are, first, the ability to give the L-DOPA through a tube that's placed through the stomach wall, through the stomach into the beginning of the small intestine, the duodenum, and that can be given by a pump. It's called DUOPA, or L-DOPA intestinal suspension. And people who take that can have as improvement in their on and off that's comparable to what they can get with deep brain stimulation. The disadvantage, of course, is you have to have a small procedure to place the hole in your abdominal wall. You have to have a plastic tube going from the pump, which you wear on your belt or carry in your pocket, and you have to fill that pump every day. But for some people, that's been really a nice step forward. Another new drug that came out in 2015 was the Ritari, which is a combination of fast-acting and slow-release levodopa, and it can help a lot with on and off. It's much more expensive than the traditional kinds, and it requires a few weeks or months of adjustment to get it just right. And then another drug that came out this year called safinamide is an exciting research advance because it helps with the motor parts of Parkinson's but makes the dyskinesia better. So continued drug development is one area of research that I think is worthwhile. There's another drug on the horizon. It's being used in Europe already. There's a drug called apomorphine, which is a relative of the dopamine agonist drugs. Those dopamine agonist drugs are things like ropinirole or pramipexole. Apomorphine has been around for a long time as something you can inject once to turn on very quickly, and now we think it's going to be given by a pump where there's a plastic tube underneath your skin and giving it to you continually. That's been successful in Europe, and we expect it to be released in the United States sometime in the next year or two. So everyone who lives with Parkinson's know that the motor parts of Parkinson's are just part of the problem. And there are a number of other parts of Parkinson's that we need to do research on, because helping those parts of Parkinson's can help you live with Parkinson's a lot better. One problem is dropping blood pressure when you stand up. That's called orthostatic hypotension. For some people, that can be very debilitating and can decrease their ability to walk even more than the stiffness and slowness of the Parkinson's does. There's a new drug for that called droxidopa. Droxidopa is a relative of norepinephrine or noradrenaline. In a way, droxidopa becomes norepinephrine in the brain just the way levodopa becomes dopamine in the brain. And for some people, that's very effective for the low blood pressure. Another challenge that people have late in Parkinson's is starting to have hallucinations or delusional thinking, and there's a new drug for that called pimavanserin that came out in 2016, and that, for the people who need it, is a really terrific step forward. So it's not true that the pharmacologic solutions are not year by year making life better for people with Parkinson's. But having said that, I want to get to the more scientific material and try to talk about some of the research that I think in the next few years is going to lead to ways to cure or prevent Parkinson's disease. And that's the story that starts with a family that lived in a town in Italy called Contursi. That's in southern Italy. and they were first described in the medical literature in the late 1990s. And in that family, Parkinson's disease was an autosomal dominant disease. That means that if you had the disease, about half your children would get it. And if you can look at this family tree that I've shown on the screen, it's a large family. To the left is the whole family tree, and I've just enlarged a small part of it to make it more visible. So black are people who have Parkinson's, and yellow are people who don't. And the circles are women, and the squares are men. So the characteristic of this is that if you have the gene, about half the people in the next generation get it, and they can happen to men or women. That's called an autosomal dominant pattern. Now I should say that inherited Parkinson's disease is still the minority of Parkinson's disease. And this particular gene, which is now called the PARK1 gene, accounts for less than 1 in 100 people with Parkinson's disease. But this genetic discovery has led to an incredible breakthrough in research that I'm going to spend some time talking about. I put an arrow by one person in the family. That's a man whose mom has Parkinson's, his sister has Parkinson's, and 4 of his 9 children have Parkinson's, but he never came down with it. No one knows why that was. Is it because he died too young? Is it because he had another gene in his body that protected him? Is it because he was taking some other element in his environment that was preventing it? We don't know. When there's a genetic disease and it skips an occasional person who must carry the gene, we call that variable penetrance. So once this gene was discovered, people in the laboratory were able to identify which gene it was and what protein it made, and the protein that gene makes is called alpha-synuclein. And you're going to hear me use that word alpha-synuclein a lot as I go on. When one of the pathological definitions of Parkinson's is that people with Parkinson's, if we look at their brain after they die, they have extra material inside their brain, what's called inclusions in their brain, called Lewy bodies. And those Lewy bodies are a very characteristic part of Parkinson's disease. And one of the things that's important when people looked at the people from Contursi who had the PARK1 gene is that their brains also had Lewy bodies in them. Some kinds of inherited Parkinsonism do not have Lewy bodies, but we're going to be talking today about kinds of Parkinsonism, inherited or not, that do have Lewy bodies. So after the gene was discovered and the protein was discovered, researchers made antibodies against the protein alpha-synuclein. They took slices of brain. They washed the slices of people with Parkinson's. They washed the slices of brain with the antibodies. The antibodies were connected to stains, so any place the antibody went turned brown. And in this slide, you can see that with that stain, they show that the Lewy bodies were full of alpha-synuclein. So, and that was in people who did not have the bad gene. In people who have regular idiopathic Parkinson's disease, have normal genes, and yet their Lewy bodies are full of alpha-synuclein. So, of the 4,400 articles on Parkinson's research last year, nearly a tenth of them were about alpha-synuclein. Now, one of the disadvantages of doing a webinar is I cannot tell if anyone is hissing or groaning with this slide. So you'll excuse me, but I'm going to talk about fruit flies for a minute and how they're used in research. What people can do with fruit flies is take an extra gene and put it into the fly genome. So fruit flies usually don't make alpha-synuclein, but if you give them a human alpha-synuclein gene, you can see if it has any effect on them. In this experiment, the fruit flies some had their normal genes, some had an added normal alpha-synuclein gene, and some had an abnormal alpha-synuclein gene, the kind of gene that was in the family from Contursi. Fruit flies live up to a little bit less than 2 months, and in the last couple weeks of their life, they start to be less active, their motor abilities fade. And the way you test that is you put the fruit flies in a test tube, and you shake the test tube so that they all fall to the bottom, and see how long it takes them to climb up. And it turns out that when they're young fruit flies, if they have alpha-synuclein, look normal, but prematurely they lose their ability to climb. They get a motor deficit. They get the fly equivalent of bradykinesia, if you will. And that happens to them whether they have the mutant gene or whether they have the normal gene. So that something about alpha-synuclein can affect motor function as the fruit fly brain gets older and as human brain gets older. So what has been done now experimentally is if we take Lewy bodies from people who died with Parkinson's disease, we know they're full of alpha-synuclein. In mice or in monkeys, you can inject that into the part of the brain where Parkinson's pathology usually develops. That's in what's called the substantia nigra. So in this, if you read the science of this, it talks about intranigral. That means in the substantia nigra. Or intrastriatal, that's in the striatum, in the putamen, where the Parkinson's cells where the dopamine cells end up. And if you inject alpha-synuclein-rich material into those parts of a mouse or monkey brain, that material spreads in the neurons, and in those animals starts to cause an illness like Parkinson's disease. So that's evidence that the alpha-synuclein is toxic in the brain and can spread in the brain. And that's really sparked all sorts of research about ways to stop the alpha-synuclein from developing and spreading. So everybody has alpha-synuclein in their brain, and all alpha-synuclein is not bad. But in this hypothesis from an article by Alano and Prusner is that for some people, the good alpha-synuclein, the one on the left, I apologize for those of you who are green-red colorblind. On the circle is a green circle and the square is a red square. So in some people, the good green alpha-synuclein gets changed to bad red alpha-synuclein. We think that's because an extra phosphate group is added to it. And then once you've got a bad red alpha-synuclein, it can start to bind together into fibrils, small fibers, and those eventually form the Lewy bodies. And we think that this misfolding of the alpha-synuclein, this formation of these fibrils, is part of the pathology that leads to the cell degeneration of Parkinson's disease. So the idea developed, is there a way we could get rid of that bad alpha-synuclein by making antibodies that would grab hold of it and take it out of the body? And a couple experiments have been done to try to do that. The first experiment was done with a product called Affitope. And the idea of Affitope is that you inject this under people's skin or as a vaccine, like you give them an immunization, and that causes people to make their own antibodies against alpha-synuclein, and then those antibodies of alpha-synuclein will get rid of the bad alpha-synuclein in the brain. You'll see on the slide that I've given you a web address, www.clinicaltrials.gov. And you might write that down if you don't know it already. I'll show it on a number of subsequent slides. That's a terrific resource because all experiments in humans, therapeutic experiments in humans, are supposed to be labeled on that website. And if you want to know what the latest research is on Parkinson's, go to www.clinicaltrials.gov, type in Parkinson's, and you'll get a whole list of experiments that are currently going on. So it's a way, as time goes on, for you to learn about new experiments that I haven't told you about. So in the trial of Affitope, there's been a phase 1 trial that shows that it is safe to have a vaccine against alpha-synuclein. Phase 1 means they've just given small doses and they're just trying to check its safety and looking for the right doses. So it's very early in development, but one approach to this getting rid of alpha-synuclein by making extra antibodies. Another way to do that is to make what's called a monoclonal antibody, which you then give to people intravenously. An antibody is a protein. That means it's a large molecule. If you swallowed it, it would get digested and wouldn't get into the body. That's why it has to be given as an injection. So the idea is to inject the antibody. The antibody goes through your blood, picks up all the alpha-synuclein it can, and helps your body get rid of it. And this is now going into larger trials, what are called randomized double-blind trials in patients with early Parkinson's. They want people in these trials to have not yet started L-DOPA. So OHSU is part of this trial and is going to be recruiting for people who want to participate. And I've put down the contact number and address of the study coordinator if you're interested in participating. They're going to use a monoclonal antibody against alpha-synuclein. It's going to be given by vein once a month. People who are eligible for the trial have to have stage 1 or 2 Parkinson's. Stage 1 is when it's unilateral. Stage 2 is when it's become bilateral, but a person is still quite independent. And people to enter the trial have had to never taken levodopa. People entering the trial, 1/3 are going to get low dose, 1/3 are going to get a high dose of the monoclonal antibody, and 1/3 are going to get placebo, and it's going to go on for a year. So going into this trial, if any of you meet these criteria and want to participate, I would love to have you volunteer. We need more people with Parkinson's helping out with all the research that has to be done. But we have to go in with the realization this is an early trial. We have no idea whether 52 weeks is long enough for it to show up. We have no idea if the dose is right. We have no idea whether, even though it's starting in early Parkinson's disease, whether to really work a treatment like this might have to start earlier before a person has the motor manifestations at all. But it's an exciting time for this trial to be going on. As ideas about alpha-synuclein have come up, there's been as ideas about Parkinson's have come up, there's been a desire to find drugs that aren't quite as complex as a monoclonal antibody. The idea is that if we take drugs that already existed, that have already been approved for something else by the FDA, that are smaller molecules that you can swallow and get into your body, could we, in experimental systems, screen those tens of thousands of known molecules that meet those criteria? Could we screen them and find some that would work on change alpha-synuclein in some way? And I'm going to tell you about 3 ideas that are going on about that and are in or near clinical trials. To do that, I'm going to talk about another genetic disease that affects Parkinson's, and that is Gaucher's disease. Someone in the audience has asked, is it true that young onset Parkinson's is more likely to be genetic? And that is true, but those young onset genetic Parkinson's don't often have Lewy bodies, and so they probably don't get treated the same way the older onset does with these new therapies. So among Ashkenazic Jews from Northern Europe, there are a number of diseases that are more common, and I'm going to talk about one specifically called Gaucher's disease. It's a recessive disease. That means to have Gaucher's, you have to get a bad gene from your mom and a bad gene from your dad, and the gene for that is called GBA, which is short for the gene for an enzyme called glucocerebrosidase. That illness varies quite a bit. Some people who have the disease are very sick in childhood. Others can have the gene and never be sick. And typically, people who are sick with it have a big liver, a big spleen, they get anemic, and some of them get neurologic diseases. As people have learned to treat this disease, enzyme replacement therapy was developed a few years ago, and that means that because people who had 2 bad genes couldn't make the enzyme, if you gave them shots of the enzyme, they would be protected from the disease. And so clinics developed where many people with Gaucher's would come together, they'd be given the enzyme replacement therapy, and as people were watched in those clinics, it became apparent that some of them developed Parkinson's disease even as they were living longer. So researchers in Israel did a study, and the Dr. Rosenbaum in this study is no relative of, unfortunately. They did a study in Israel, and they looked at people with Parkinson's disease in Israel who were not known to have any genetic cause. And they looked at 3 groups of people: people who didn't have Parkinson's, people who had Alzheimer's and people who had Parkinson's. In the normal population among the Ashkenazic Jews in Israel, 6%, about 1 in 17, carries one copy of this gene. They don't have Gaucher's disease because they only have one bad copy. People with Alzheimer's have no more incident of that gene than other people. But people with Parkinson's, nearly a third carry that bad gene. So there's something about carrying that gene that makes people more susceptible to Parkinson's. And it now turns out that this mutation, the GBA mutation, is the most common genetic risk factor for Parkinson's disease. If you have that one copy of that gene, you're 20 times more likely to get Parkinson's disease. And if we take all people with Parkinson's, this applies not just to the Ashkenazic Jews, but all people with Parkinson's all over the world, 5 to 10% of people with Parkinson's have this mutation. It's a mutation with what's called variable penetrance, which means most people who get the mutation do not get the disease. And by age 70, between 5 and 7% have it, and by age 80, 9 and 12% have it. So it's a risk factor. By itself is not sufficient to cause it. So people have looked at people with the GBA mutation who have Parkinson's, and they look just like anybody else with regular Parkinson's disease. They have the same non-motor symptoms, loss of smell, constipation. They have the same motor symptoms, the tremor, the stiffness. They're a little bit younger when they get it on average, but not much. They're a little bit more likely to have trouble with their thinking, but not much. They respond to medications the same way, and when they die, their brains have Lewy bodies. So it's a very close relative of idiopathic Parkinson's disease. So people have looked, using fruit flies again and other experimental models, have looked at why the GBA mutation causes Parkinson's. And the thing that's really interesting is that if you don't have the GBA in your body, if you don't make the enzyme, at least in fruit flies, you make more alpha-synuclein. So it looks now like there's a system in the brain. The enzyme I'm going to abbreviate now is GCase. The glucose cerebrosidase I'm abbreviating as GCase. The less GCase you have in the brain, the more alpha-synuclein you have in your brain. So is there a way we can get a drug that would increase your GCase and that would get your alpha-synuclein down? And it turns out there is a drug that can do that. And the drug was discovered from an old cough remedy called Ambroxol, which is not sold in the United States, but it is sold in Europe as a cough syrup. You can take it as a syrup or a tablet. It's good for coughs with thick mucus, and some people think it helps sore throats. It has relatively few side effects like upset stomach. Very rarely it can cause Stevens-Johnson syndrome, which is a very serious skin rash. But it was found that this commonly used cough syrup, when given to mice, decreases their brain alpha-synuclein, and the same thing is true in non-human primates. So at the University of London right this year, there's a trial in progress giving Ambroxol to people with Parkinson's disease. They're using doses that are quite a bit higher than people would take for cough. So typical cough dose is 60 or 120 milligrams a day. The people in the Ambroxol trial are going to get anywhere from 180 milligrams a day to over 1,000 milligrams a day. Some of the people in the trial are going to be GBA mutation people who are low in GCase, and some are going to be people without the mutation who have normal GCase. And the trial is going to be looking at the blood and spinal fluid to see if Ambroxol can really increase people's GCase levels. So this is a very preliminary trial. It's a long way from showing you can increase a person's GCase level to actually show that you slow the progression of their Parkinson's disease. But it's a very creative idea. And I learned about it at the last World Parkinson's Congress in September 2016 in Portland, when Dr. Shapira from London gave a wonderful hour talk about this. So I'm sorry I only can tell you about it for a few minutes, but I think it's a really exciting idea. So another idea is it a way to speed up the way the brain gets rid of alpha-synuclein? In your cells, you've got to have systems to get rid of bad proteins. And those systems include something called proteasomes and something called autophagy. And is there a drug that would increase that garbage system to get rid of the bad protein? And it turns out there is a drug called nilotinib that does that. Now, nilotinib was first developed to treat leukemia. Maybe some of you have heard of Gleevec or imatinib. Any drug that ends in the words TINIB is what's called a tyrosine kinase inhibitor. And the first one was imatinib. It's terrific treatment for certain kinds of leukemia, used for a few other kinds of cancer. But it turns out that nilotinib also increases cellular disposal of alpha-synuclein. And in a small study that was done at Georgetown and reported last year, a few patients taking nilotinib seemed to have improvement in their Parkinson's, including improvement in their thinking. Now, nilotinib is a very heavy hitter drug. It's got a lot of very serious side effects. It's a very expensive drug. It costs $125 a pill, and people with leukemia take $500 worth a day. So what's going on now in two different trials, one at Georgetown and another at a number of other centers, is that people are trying lower doses of nilotinib to see whether the lower dose can be given safely and whether it might have an anti-Parkinsonian effect. You can go to www.clinicaltrials.gov to find out where those trials are being done. I think the nearest one in the Northwest has been doing by Jason Aldred in Spokane, and I don't know whether he's still recruiting patients or not. So last month, one of my patients brought me in an article with this headline, which was from Parkinson's News Today. And it said that people at Michigan State are doing research and using a common antidepressant to delay Parkinson's progression. Again, a small molecule, you can swallow it, and it can get into the brain. And they've shown that drugs in this class, they're called tricyclic antidepressants. One's nortriptyline, one is nortriptyline, and another one is amitriptyline. And it turns out that nortriptyline in cell systems blocks the change from a single bad alpha-synuclein molecule to a bunch of bad alpha-synuclein molecules. And that was shown a few years ago. That was shown recently. Now, there is human reason to believe that this might be helpful because a study that was done in Norway, excuse me, I'm confusing my studies. This is not the Norway study. Pommier and his colleagues looked at a number of studies that had been done to treat Parkinson's disease. And they went back and looked at people in those studies, and they looked whether the ones who were taking nortriptyline and amitriptyline were any different in their chance of coming down with Parkinson's than people who weren't. And it turned out that people who were taking nortriptyline or amitriptyline waited longer before they needed L-DOPA. Now, the tricyclics have been around for many years. They were the antidepressants of choice before we got things like Prozac and the other SSRIs. But when the newer antidepressants came out, the SSRIs came out, the tricyclics fell into disuse because they have a lot of side effects called anticholinergic side effects. That means they cause constipation, dry mouth, they can cause memory loss, they can lead to weight gain, and they really were used much less often for depression than they used to be. If anything, we use them nowadays more to treat migraine and to treat chronic pain. Now, anticholinergic is a pharmacologic term. It means it blocks the neurotransmitter acetylcholine, and before levodopa was around, anticholinergic drugs were the best drugs to treat Parkinson's. So you can ask if the tricyclics are anticholinergic and the anticholinergics treat Parkinson's, is the tricyclic treating the Parkinson's directly, or is it really somehow being preventive? And the Pommier and his colleagues in the study went back and showed that it looked like it was not making the Parkinson's symptoms better, and so they are hoping that it might be preventive. So my patient said, well, you prescribe tricyclics all the time for people with migraine and pain. Why don't you just give me some? And I said, well, it will make your constipation worse. We almost never give to people over 70 because it bothers their memory. And after talking about it with that particular patient, we decided not to try it. But it's an interesting idea for another way to change alpha-synuclein accumulates in Lewy bodies. The next idea is there a drug that could just take the gene that makes the alpha-synuclein and turn it off? And it turns out that there's a small molecule that does that, and it's called salbutamol. Salbutamol is a drug that's commonly used for asthma. In the United States, it's often called albuterol, and it's commonly used in asthma inhalers. And research is showing now that this drug stimulates the chromosome in a way that cuts down the production of alpha-synuclein. So that's known in experimental situations, and the human evidence comes from an 11-year study of 4 million Norwegians, and it turned out those who had ever used salbutamol were 1/3 less likely to get Parkinson's than those who had never used it. It also turned out that if they'd used a beta blocker drug like propranolol, which does the opposite of salbutamol, they're more likely to get Parkinson's. That's a little bit of a debatable observation because sometimes we give propranolol for tremor, and it might be the people were taking propranolol for tremor and they already were Parkinson's prone. So I don't know if we can make too many conclusions about that, but there's been a lot of interest in salbutamol as a new drug to try either to prevent Parkinson's or to keep it from getting worse. So this is very much in the infancy. It just came out in September, and one of the researchers involved said that so far there have been 16 other drugs tried to prevent Parkinson's. They've all failed. I hope we're not the 17th. So I'm going to leave drugs in addition in a minute. And before I do that, I'm going to answer a couple of questions that have been asked. Someone asked, is ilotinib generic yet? I don't think so. I don't think it's generic. And even if it were generic, our experience with these very expensive drugs, that wouldn't decrease the price by very much, and we'd still have a lot of side effects to worry about. So I don't think ilotinib is a thing that people should gamble on just in case. I think if you're interested in ilotinib, try to look on www.clinicaltrials.gov and see if you can be accepted into one of the experiments. Another person asked, are these drugs in addition to L-DOPA or in place of? All the things I've been talking about in this experimental section are drugs that we hope will prevent or slow progression of Parkinson's, and would therefore you start them before you started L-DOPA, or take them in addition to L-DOPA, and whether or not you needed L-DOPA would be a separate question. So stem cells is a separate issue, and I'm going to talk about it for a few minutes. Earlier this year, I spent a whole hour talking about stem cells, and at the end of the stem cell question, all the questions were about other drugs, and that's how come this whole hour is not about stem cells. But anyway, stem cells get people really excited. If you go on the internet and look up Parkinson's disease and stem cells, you'll get all sorts of advertisements, and be very wary of those advertisements. If people are advertising it and want to charge you money for it, the chances are it's not research and it's not approved yet. And one of the reasons that I'm particularly wary of following one of those ads is this story of a 66-year-old man who was a stem cell tourist. He went around the world getting people to give him stem cells to try to reverse the effects of a stroke. He got stem cells, paid for stem cells in China and Argentina and Mexico. By the time he was done, the stem cells, which had been infused into his spinal fluid, had caused a tumor that was pressing on his spinal cord. So I wouldn't go into commercial stem cell experiments yet by any means. But there are a number of studies going on now to look at different kinds of stem cells for Parkinson's. One is being done in Melbourne, Australia, and it's using 3 different doses. And whether or not it's the right thing to do is debatable. I've given you a reference at the bottom of the slide to Dr. Barker, who believes it's the wrong thing to do, and you can read why he doesn't think this is a good trial to get involved in. Dr. Barker and his colleagues in Europe have another trial going on where rather than giving stem cells, they're putting in fetal tissue taken from the midbrain. That's tissue that's rich in that's tissue that's rich in dopamine cells, and they're implanting it. This is the same kind of treatment that was done in the United States a number of years ago and had a negative trial, but the Europeans believe they've improved their system so that they're now going to be able to have a successful trial. And they're recruiting patients, and they hope to be done with their trial by 2020. People going into this study are being started relatively early in the disease. I myself am a little bit dubious about these kind of implant treatments as really the solution to Parkinson's disease. And the reason is that, Parkinson's affects many parts of the brain. It affects the loss of smell, it affects the GI system, it affects sleep, it affects the skin. And each of those areas are different kinds of cells, different neurotransmitters, and just putting stem cells into one spot in the brain to make more dopamine is not going to solve the whole problem. So trying to get the cells that give the right neurotransmitter, go into the brain, survive, don't cause problems like tumors, can adjust themselves to become part of brain circuits is going to be a real challenge. And one thing that's been very interesting among the people who years ago did have fetal cells placed in their brain to treat Parkinson's is that we've learned that the Lewy body pathology, the alpha-synuclein pathology, spreads into the new cells. So that getting into the new cells doesn't solve the main problem. I think alpha-synuclein is the way to go, and stem cells is not as exciting. There is another stem cell system being done in China where they're giving the stem cells in the nose, which is where the smell problem starts. So I've talked about the multiple parts of Parkinson's disease. I've talked about the importance of alpha-synuclein, and I want to now take a slightly different view and talk a little bit about the gut in Parkinson's disease. We think that Parkinson's starts either in the nose or the gut and slowly spreads through the brain and only gets to cause the tremor and the rigidity after it's been brewing in the body for 5 or 10 years or more. And one of the earliest places that the Parkinson's pathology develops is in the gut. Your gut has as many nerve cells as your brain does. And there's a nerve called the vagus nerve, which connects the nerve cells in the gut to the part of the brain called the medulla. And so we think that Parkinson's may start in the gut, spread to the medulla, and then spread elsewhere in the brain. So people have been very interested in looking at the bacteria and the other organisms that live in the gut, what's called the gut microbiome, and to see if that has anything to do with Parkinson's disease. You have more genes, more different genes in your microbiome in your gut than you have in human genes in your body. So this experiment, which was published last year, is a really interesting story. They took mice and they genetically modified the mice so that they were making alpha-synuclein. So that made the mice susceptible to develop an illness like Parkinson's disease. And they raised some of the mice germ-free so that the mice, if the mice led a normal life and had no change in their usual gut bacteria, they would develop a motor disease like Parkinson's disease. But if they raised the mice germ-free, and when they were born, they put them in an environment where they couldn't get infected with anything, so their gut bacteria did not develop, they never developed the motor aspects of the Parkinson's. Or if they let the mice get infected with germs when they were babies, but then gave them antibiotics to get rid of the bacteria from the gut. They're what's called non-absorbable antibiotics that they can put in their water. They drink it, they get the antibiotic, it kills the gut bacteria but doesn't get into the body. Those mice also didn't get the motor deficits. They also showed that if they took a kind of chemical called short-chain fatty acids that are made by those gut bacteria and injected them into people, that could also lead to getting the motor deficit. So there's the idea that something happens in the gut, something happens with what's infected in all of our guts that contributes to the development of Parkinson's. And as part of the experiment, they took these mice, the ones who didn't have normal bacteria, and they took some bacteria from the guts of people who actually had Parkinson's disease. They fed that to the mice, and the mice got Parkinsonism by being exposed to people's Parkinson's bacteria. So the idea that the microbiome might be really important about who develops Parkinson's disease why is a very exciting idea. People have looked at the bacteria in people with Parkinson's disease, and it turns out that people with Parkinson's disease have different bacteria in their gut than other people do. This is true whether or not they've started L-DOPA therapy or not. In fact, they already have these abnormal gut bacteria before they get the motor system symptoms. So you may recall, you may know that there's a sleep disorder called REM behavioral disorder that in about half of people with Parkinson's comes on a number of years, 5 or 10 years before they get any motor impairment. And people with REM behavioral disorder already have the change in their gut microbiome that goes with eventually developing Parkinson's. So should you get a stool sample and send it to one of the people, look on the web, you can find people who will look at your gut microbiome and tell you whether it's normal or not normal. And this article in the New York Times this summer quotes experts as saying, you'll learn a lot about what's in your gut, but it won't tell you what to do for treatment, and it's really an area that's quite in its infancy, but a very exciting area and one that I think is going to lead to very important insights and new treatments in the future. So I have a personal interest in this. My dad had Parkinson's, my uncle had Parkinson's. That means that I'm at least 5 times more likely than other people to get Parkinson's. And I'm wondering, should I be doing things to prevent Parkinson's? I wouldn't go near the lotinib, too expensive and too dangerous. But ambroxol, I might be able to find some, have a friend in Europe mail it to me. Nortriptyline, I probably can tell my doctor that I have migraines, and maybe he'd give me some. Salbutamol, if I have a little bronchitis, I might be able to get some from my doctor. And I could take a probiotic, maybe some extra yogurt or something. Should I be doing all these things? If I do each of these things to decrease the process of having synuclein become toxic in my brain, do I decrease my risk of Parkinson's? My personal view is that I'm not going to do anything like that, that this is very exciting but very preliminary research, and it's not yet time for any of us to start doing it to ourselves, but it is time for us to volunteer for research projects that may lead to learn whether it's working or not. So if you want to learn more about this, I've already mentioned clinicaltrials.gov. I've listed 3 websites that are pretty good at bringing out the headlines and what's new in research. But I'll warn you, as you look at these headlines, that people get excited, like the story about the salbutamol, and you got to go look behind the scenes to find out exactly how close it is to being ready for people to take. So I would urge you, if you hear about some new research, to talk to your own physician to think about the pros and cons of it and where it might lead and whether it has any relevance to you. So I've been very fast. I've taken the liberty of being relatively scientific, because part of the concept that came from the World Parkinson's Congress is that people with Parkinson's and the researchers should try their best to talk to each other and speak the same language. But as I said, there's not going to be a quiz for you, but there is a quiz for me. So please send Libby some of your questions, and I'll see if I can make things any clearer. Thank you very much. So people have asked, are the slides available? I will create a handout, and if you write to Pro, we can figure out a way to get you access to it. We'll put it on the Pro website or do something like that. So someone asked, is OHSU the only local place to get involved in studies? At our Portland Parkinson's program, we have been involved in studies at time. We don't have anyone going on right now. The best way to find out is when you go to clinicaltrials.gov, it will tell you all the people in the country who are doing it. Someone asked about nicotine and Parkinson's disease. I don't have anything new to say about that today, I'm afraid. So questions are being typed in, and so that's why we have a bit of a pause.
Can you hear me now?
Yes, I do hear you.
Okay. So are you seeing the questions? I think that everyone might be hearing me. Are you seeing the questions I've sent over to you?
The last one I saw was the one about nicotine. Are there more since then? Oh, I'm sorry, I haven't. Okay. Excuse me. Okay. So thank you, Libby. What effect does alcohol have on Parkinson's disease? There's no major effect that we're aware of. The once in a while can decrease the tremor a little bit, but has much more effect on essential tremor than on Parkinson's tremor. In general, I neither encourage nor discourage use of alcohol among people with Parkinson's. I wouldn't want to take more than 2 drinks a day for sure, because that increases your risk of stroke and of psychiatric, not psychiatric, but rather cognitive problems. Next question is, exercise, a hot topic. What's best? Exercise is a very important topic. We think that it has ability to improve or possibly prevent Parkinson's. We don't know the dose. So I tell people a few things about that. One is exercise for at least 30 minutes 5 times a week. Of course, that's the recommendation for everybody. The second is do a kind of exercise that you enjoy. The third is vary your exercise. So it's really nice to do a balanced exercise like Tai Chi or yoga. It's good to do some aerobic exercise. On the Pro website, there's a list of a number of different exercise options, and I would encourage people to try a number of them and find the ones that they enjoy the most. Next person asks, if my symptoms don't respond to L-DOPA, might that indicate that I don't actually have Parkinson's? Well, about 10% of people who look like they have Parkinson's don't respond to L-DOPA. And sometimes that means that the diagnosis is incorrect, that they have a relative of Parkinson's, what's called Parkinson's plus or Parkinsonian syndrome that's not really idiopathic Parkinson's disease. So that's a very good question, and it's something to talk to your own doctor about whether you've had an adequate trial of the drug and whether there's an alternative diagnosis. So does exercise really change the disease, or does it just make me tired? No, we think it really changes. We think it really changes the disease, and so we think that it makes the brain have better connections and better synapses. We think it's really good for the brain. So get that exercise in. Cannabis liquid under the tongue? Question mark. The CBD oil, or the cannabis liquid, it is not completely free of the part of the cannabis that can cause mental changes. People have to, there's no proof of whether it's good or bad. I would say that people who want to try it should try it. Not keep it a secret from their doctor, but talk it over with their doctor, give it a try, and let the doctor know whether it helps or not. What about SSRI antidepressants? What about Paxil with carbidopa, levodopa together? It's a combination we use commonly because people with Parkinson's often have depression as part of their Parkinson's, and I'm not aware that it has any effect on the long-term course of the Parkinson's. Someone asked, agonists seem such a problem for some people. Your thoughts on risk versus benefits? Agonists have advantages and disadvantages. We often use them in combination with L-DOPA. Some neurologists give them as the very first drug before L-DOPA. They do have other side effects like confusion, hallucinations, low blood pressure, impulse control disorders. So they've got to be adjusted very carefully with your doctor's help. Is there anything new to control postural hypotension? I mentioned the droxidopa, the Durthera. That's the newest thing we have for that. Next, someone asks, how should one go about getting tested for the PARK1 gene? My dad and 2 of his first cousins have Parkinson's disease. I am not encouraging my patients to get genetic testing for therapy because I don't have any new therapy based upon the gene. Families that do have Parkinson's disease can really participate in research. There's a genetic trial going on at the University of Washington, and if you wanted to participate in their research on genetics of Parkinson's, I'd encourage you to contact the Department of Neurology there to ask about participation. But I don't think that there's a role for testing for the gene right now. I think that could change any year as soon as we find one gene that has a different treatment. And when that happens, we're going to start doing genetic testing, but not until then. Someone asked about diet and nutrition, and what about diet? What about losing weight? Well, the first thing I'd say is that there's no food that's known to be bad for Parkinson's disease. When you're taking levodopa, the amount of protein in your diet is an important issue that you should talk about with your doctor, but it's usually an issue that's easily dealt with. Should you try to lose weight when you get Parkinson's? As Parkinson's progresses, people often lose more weight than they want to lose because their stomach doesn't empty well, they don't eat as well. So I wouldn't go overboard on weight loss if I had Parkinson's disease. And in terms of diet, I would eat a heart healthy diet. A speech pathologist writes about people who have swallowing trouble, and that the swallowing problems can fluctuate during the day. It's certainly true that on-off can affect the swallowing, but I think that's one thing that's really important with speech and swallowing trouble is that they often are relatively resistant to the medications. And if you're having speech and swallowing trouble, be sure to talk to your doctor about it. Be sure to get referred to a speech and language pathologist about it, because the speech therapy can be one of the most effective things for those kinds of problems which are not as amenable to medication. So someone asked about complementary therapies, which is another exciting area, and specifically about CoQ10. 15 years ago, we were very excited about CoQ10. Unfortunately, it's turned out not to be of benefit in the more complete studies. So I had a number of people taking CoQ10 for a decade, and I'm not urging them to spend money on it now. Someone asked, are there any studies about alpha-synuclein for patients who have been on levodopa? I don't know that, and that's a question to look at on clinicaltrials.gov. I'm not aware of one right now. I mentioned the monoclonal antibody study that's being done at University OHSU. There's another monoclonal antibody study that's being done by another drug company, and the details are on clinicaltrials.gov, and you can see who they're accepting for their patients. So we're right at the end of the hour. You've told me that I'm too fast, but I've had to talk fast to get it all in, and there's even more to say, but I'm afraid I'm going to stop right now. Thanks a lot.
Thank you, Dr. Rosenbaum. Okay, so this webinar has been recorded, and we will have it up soon. There'll be a link on our Facebook page. So thank you. We got through lots of questions. It was a lot of information, and I think that I myself will have to watch this over again because I was busy taking care of the webinar, and I want to go back and pay more attention to all this exciting information. So thank you so much, and watch for future webinars that we will have. All right. Bye-bye.
We're off.
Yeah, I'm going to close.